What do you do if you need to study a native protein, but you don't have specific antibodies? LabSurvivalGuide is back. Today I'll walk you through the fundamentals of adding protein tags like MEK, HIS, FLAG, HIBIT, and others. It's a fundamental technique in biochemistry, molecular biology, and cell biology. It is used to overcome challenges in protein expression, localization, purification, and detection, especially when specific antibodies are unavailable. Getting it right can make or break your experiment. Let's dive in. A protein tag, also called an affinity or epitope tag, is a short peptide sequence, usually 6 to 20 amino acids. In order to work, it needs to be fused to your protein of interest. As mentioned, the main reason for using a protein tag is to help you study your protein more easily, using commercial reagents that are already validated. But that's not the only reason. Some tags, like MBP or SUMO, are added to help the protein fold properly, stay soluble, and avoid aggregating into inclusion bodies. Just keep in mind, these tags can be pretty large. Since you do not want to disrupt the protein's native structure or function, choosing whether to place the tag at the N-terminal or the C-terminal, it's a matter of biology. Start by checking the literature or databases. If the N-terminal contains signal peptides, membrane targeting sequences, or catalytic domains, choose C-terminal tagging. If the C-terminal is involved in dimerization, localization signals, or is structurally buried, choose N-terminal tagging. Some proteins tolerate both. Others lose activity entirely. Also remember, tags can affect folding, stability, solubility, aggregation, and immunogenicity. Never assume a tag is neutral. Your tag must be fused in frame with the coding sequence. The ribosome reads mRNA in triplets, starting at AUG and continuing until the stop codon. So the entire tag plus protein need to form one continuous open reading frame. If the number of nucleotides added between the start codon and the first codon of your tag isn't a multiple of 3, you create a frame shift, resulting in either a meaningless peptide or premature termination. Always verify the reading frame before starting the cloning. How do you modify the DNA? It depends on your cloning strategy. You can design oligos to add a tag by PCR. For N-terminal tagging, the forward primer starts with 1. ATG start codon. 2. Tag codons plus a flexible linker. 3. 18 to 25 nucleotides of your gene's coding sequence without the original ATG. For C-terminal tagging, the reverse primer starts with 1. Stop codon. 2. Tag plus flexible linker. 3. 18 to 25 nucleotides of your gene without the stop codon, all in reverse complementary orientation. You can also use commercial vectors which already contain an in-frame tag next to the multiple cloning site. In this case, remove the ATG if the tag is N-terminal. Remove the stop codon if the tag is C-terminal. Always confirm the final sequence in silico. In genome editing approaches, tags are usually added at the C-terminus of the gene. The donor template that carries the tag is often a single-stranded oligo of 100 to 200 bases. The tag sequence is flanked by 40 to 60 base homology arms that match the genomic region around the cut site. Be sure to introduce silent mutations in the PAM or guide seed region, otherwise, Cas9 may cut your edited allele again. Also confirm that the tag does not disrupt splicing or regulatory elements. To prevent steric clashes and improves accessibility for antibodies or affinity beads, always include a linker between the tag and the protein. A short glycine serine repeat such as GSGSG, up to 6 repeats, is common, but for better accessibility, especially with bead-based purification, the linker can be extended up to 15 repeats. If you want a protein as close as possible to its native state, after purification you can add a protease cleavage site between the tag and the protein. Common choices are TEV, typically leaves a single glycine. HRV3C, leaves glycine proline. Thrombin, leaves glycine serine. Factor XA and Enterokinase, both remove the tag without leaving extra residues. Choose based on your downstream application. Additional practical tips. Codon optimize for your host to avoid rare tRNA limitations. Avoid overtagging. Multiple tags can significantly reduce expression yield. Always validate function after tagging using untagged version, where possible. Recap. A protein tags are powerful tools when specific reagents are unavailable. Choose the correct terminal location. Maintain the reading frame. Include flexible linkers. Optimize codons. Avoid overtagging. Use protease sites when necessary. Design carefully, and your tag will work for you, not against you. See you soon.